Oh, fantastic. Okay, it looks like we are officially live. Hi, everyone, and welcome into the Seymour Marine Discovery Center's live program today. It's so great to see everyone settling in. Yes, you are early. We are starting early. I shouldn't say start. I should just say getting folks settled in, giving folks the opportunity to get really excited for this upcoming Labside Chat program. It is 10:58 over here, so we have about 2 minutes until we officially start this conversation. But as folks are coming into this Labside Chat, go ahead and say hi in the live comments. We'd love to see where our viewers are watching from, and go ahead and let us know. And maybe even Carl and I can give you a shout out during our conversation today too. So go ahead and say hi in the live comments, and we love to see where our supporters are tuning in from. Oh, this is very exciting. Hi, everyone. I wish we could see you so much. We miss seeing all of the Seymour Center supporters, but it's so great to see everyone joining in. Oh, it looks like Lexi is joining us. Hi, Lexi. It's so great to see you. But yes, if you are just joining us, we are going to get started in just about a minute. I hope you all are getting excited for this Labside Chat. But go ahead and say hi in the live comments. Like I said earlier, we love to see where our Seymour Center supporters are watching from, and maybe you'll even get a shout out too. So stay tuned. All right, fantastic. So welcome everyone. We'll get started in just about a minute. We're giving folks all the opportunity just to get settled in and get ready for today. Okay, oh, here we go. Okay, so it looks like it is almost 11 o'clock, fantastic. And we are just giving folks the opportunity to get settled in. We'll get started in just probably about another 30 seconds or so. We're giving folks the opportunity to join us. And once folks join us, we will officially get started. But if you missed it earlier, I said go ahead and say hi in the live comments. We love to see where you're watching from. Go ahead and interact with us, and we'll be able to say hi right back at you. All right, awesome. So you know what, Carl? It is just about 11 o'clock. We have a fantastic program lined up for everyone watching and tuning in today. So let's go ahead and officially get started. So hi, everyone, officially, and welcome to another Labside Chat with the Seymour Marine Discovery Center. If this is your first time tuning in for today's program, welcome. My name is Jen Cormier, and today's program is an opportunity to have a short, engaging Q&A discussion with a scientist from UC Santa Cruz. And we'll be learning all about what it means to be a scientist, what it's like out in the field, what it's like to make new discoveries. So I'm really, really excited for today. And today I am joined by the wonderful Dr. Carl Lamborg. Dr. Carl Lamborg is an associate professor of ocean sciences at UC Santa Cruz and is a leading expert on mercury in the ocean. And we'll be chatting about how it ends up in the ocean, how it gets in the atmosphere, and how it can even impact us too. So Carl, thank you so much for being here today.
Of course, it's my pleasure.
So Carl, before I start asking you questions and really diving into today's conversation, I have a big thank you that I want to send to all of our viewers tuning in today, because our viewers tuning in today actually pre-submitted questions for all of you, for you, Carl, in advance. So all of the questions that I'm going to be asking are from our community members. And so thank you to everyone who submitted those questions. However, if you didn't get the opportunity to submit a question for Carl, don't worry, you can still type in your questions in the live chat or in the live comments, and we'll be able to get to those too. So Carl, I really hope you're ready. There's so many awesome questions, and I hope we have time to get to them all, but let's just get right to it.
Great.
Okay, so I said earlier that you are a expert in mercury in the ocean, but but just to make sure that I am on the same page and I fully understand what exactly that means, can you first define what mercury is, what it looks like? Does it have a smell? Like, why is it important? Like, what is mercury?
Yeah, well, mercury is an element. It's one of those things on the periodic table, one of the squares in the periodic table. What it looks like is in the form that probably everybody's or most people are familiar with, although it's getting less and less common to see these, is that silvery liquid that's inside of thermometers. At least, folks my generation would recognize that. They're more and more banned these days, those kinds of thermometers. So maybe there's some people who've never seen it. But anyway, it's a metal in its elemental, what's called elemental form. It's a metal, and it's unusual in that it's a liquid at room temperature, unlike most metals. So it's a sort of silvery looking liquid. But that's not its only kind of chemical form that one finds in the environment. And there are a couple of other really interesting and important ones. So there's mercury in the air, there's mercury in the water, there's mercury in the soil, there's mercury in creatures like us and fish and other organisms. And the chemical forms in each one of those places is a little bit different. So the form of mercury that's in thermometers is the same kind that's most of what you can find in the atmosphere, actually. So not only is mercury a liquid, it's also really unusual for a metal in that it's kind of volatile. So it's actually present in the air as a gas. That doesn't happen with iron or some of these other kinds of metals, but it does for mercury. And then in the ocean and in waters and that kind of stuff, it's mostly as a what's called an ion. So it's a mercury atom that's lost 2 of its electrons. So it has a +2 charge, and it links up with other things that are present in the water. And then inside of creatures, mostly the form of mercury you find is something called monomethyl mercury. So it's a mercury atom that has a methyl group. In organic chemistry, a methyl group is a CH, 1 carbon and 3 hydrogens glued onto the mercury atom. So those are the sort of the 3 main important forms that you would find out there. And I don't know what they all smell like, actually.
You never had the opportunity.
That's right. Maybe you don't want to know, I think is probably the answer. So the reason why we care is that mercury is toxic. It's a pretty nasty neurotoxin. And we know this sort of most tragically from this massive poisoning event that took place over sort of a long period of time, but it was most acute in the sort of 50s and 60s in a small city in southern Japan called Minamata, where they were sort of accidentally, unknowingly dumping a lot of mercury into an embayment right next to the city. And then, as now, Japanese people eat a lot of fish. And so they were getting some of this mercury pollution back to themselves through eating local fish.
Yeah, definitely. I'm glad you said that too, because when I think of mercury in the ocean, I usually think of it getting into organisms like fish. And so usually we hear those warnings like, don't, there's tuna or swordfish or sharks have high mercury content. So can you, we got a really great question about how mercury moves through the ecosystem or how it ends up in these different types of organisms. So can you elaborate a little bit more about how mercury moves through our ecosystem and how does it bioaccumulate in animals? And if you could also define bioaccumulation too.
Sure. No, it's a great sort of fundamental question. So there's sort of 2 highly closely related terms. One is bioaccumulation and the other is biomagnification. So any chemical that is more abundant inside of living things compared to its surroundings is said to bioaccumulate. And then if that same compound gets more and more concentrated in a food web, in a food chain, if it's much more concentrated, for example, in top predator organisms than it is in plants or phytoplankton, things that are at the bottom of the food chain, then you say that you would describe that chemical as something that biomagnifies as well. So there's sort of 2, and they're of course related to something wouldn't biomagnify if it didn't bioaccumulate, but so there's there's a small distinction between those terms that's sort of technical, and maybe not super important. But anyway, so mercury, and it's the interesting part. One interesting part of the mercury story is that there's really only the methyl mercury form bioaccumulates and biomagnifies. The elemental mercury form doesn't really biomagnify and bioaccumulate, and the mercuric ion form, the +2 form, doesn't really bioaccumulate and biomagnify very much. So it's really this one particular chemical form, monomethyl mercury, and it comes in mostly, it starts, it bioaccumulates fundamentally at the bottom of food chains, we think, mostly just by diffusing into phytoplankton. Because of the chemical properties of methylmercury, it just prefers to be inside of living cells than floating around in the water instead. And then after that, it's just a matter of, because it likes to be in living things so much, zooplankton, when they eat phytoplankton, they're eating the phytoplankton to get the carbon and the nitrogen and the phosphorus and all the food stuff that's in the phytoplankton. And they mostly, and they, all organisms respire that carbon. They burn it off for energy, exhale carbon dioxide when they do that sort of thing. But they keep the mercury behind. So every time they're eating a phytoplankton, they're sort of burning off the carbon part, the yummy part, and their bodies are hanging on to, sort of unwillingly hanging on to the mercury part. So right there, after one step in a food chain, the zooplankton are going to have more mercury in them than the food that they eat, because they're accumulating all of that. They're aggregating all of the mercury from the thousands of cells that they're eating and hanging on to it themselves. And then this happens every step in the food chain. So you can go from phytoplankton, for example, have about 10,000 times more mercury inside themselves than in the water in which they live. And then you increase the mercury biomagnifies by about a factor of 10 for every level you go up in the food chain after that. So by the time you get to tuna, for example, they can have 10 million times more mercury in their tissues than in seawater, for example. So if you just have a little bit of mercury in seawater, it sort of finds its way to the worst possible place, which is the fish that we want to eat.
Right. Wow. And I'm really glad that you clarified that, because I never really fully understood how mercury in the ocean got into phytoplankton. And so the fact that it just diffuses or, comes into contact with those skin tissues, that is incredible. Wow. Okay. Well, thank you so much for explaining that and helping us do that. But I'm curious, have you always been interested in this type of research and looking at mercury in the ocean? Is this something that you knew that you always wanted to do?
I guess so. I mean, I've been doing it for a while. I sort of stumbled. I knew that when I was in college that I was interested in environmental chemistry kinds of stuff, but there wasn't really, I'm old enough that there wasn't a program on environmental chemistry or anything like that. So I sort of stumbled into it. The first sort of job I had out of college was environmental chemistry related, but more atmospheric chemistry at that point. And then I met a person there in my first job who I ended up following to do my first stint in graduate school to get a master's degree at the University of Michigan. And he just decided that doing mercury research was something that he wanted to launch, but it wasn't something that he knew a lot about at that time. Actually, it was something that not a lot of people knew a lot about at that time. So I just sort of backed into it, but it's a really interesting, it's a really interesting subject for scientists and people who are interested in environmental chemistry, because it has this part of its story is in the air, part of its story is in the water, part of its story is in the soil, part of its story is in animals. And so it's just sort of fun. You can sort of chase after it all around the world. So it's a tragic story, but it's a fun story from a scientist's point of view.
Yeah. And when you say that you're chasing it all around the world, has your research taken you to really unique locations? And what is the coolest place that you've ever been to?
Well, it's in grad school. Let's see, I did a lot of work in trying to reconstruct the history of mercury pollution around the planet over time. And that involves taking sediment cores from lakes and peat bogs. So I went to New Zealand, and I went to Nova Scotia, and I went to southeastern Alaska, and different parts of other parts of Canada. That's somewhat exotic, I guess. New Zealand was really great. And then since then, doing much more sort of hardcore oceanography kind of research, then you're really traveling to all kinds of cool spots. So most recently, the last big cruise that I went on was in the Arctic Ocean, and we went all the way to the North Pole. So that was not a place that you normally go on a cruise ship or something like that. So that was really exciting.
Yeah, definitely. And you said something about how you are studying mercury contamination with sediment cores. And so that is actually leads us to a next question that we received about how do you exactly do this type of research? Like, are you out in the field? Are you on cruises? Are you analyzing different organisms in the lab? How do you study mercury in the ocean, pretty much?
Yeah, so all of the above. So there's some components of things that involve going out to the ocean or lakes or wherever you're going to collect samples and bring them back to the lab to analyze them. But we also do lab-based experiments. So that's, I haven't done very much of this, but for example, if that first step in the bioaccumulation chain that you were asking about, how mercury gets into phytoplankton, for example, you could take then some phytoplankton and grow them in the lab and give them certain concentrations of mercury and see how fast they take it up and that sort of thing. So there's a role to be played for field work and lab work. And then also, and there's a lot of people who are doing computer modeling as well, similar to the way that people are trying to understand climate change and that sort of stuff. People are trying to figure out how mercury moves around the planet and that sort of thing. So there's a lot of different ways, but what I've done is mostly sort of field worky kinds of things in my career. And on ships in particular, what you do is it's a whole bunch of people get together. They write proposals to usually the National Science Foundation is the place you go to get money to do this work. And the ships are either owned or operated by people who are also paid by the National Science Foundation. So I should just say thank you to everybody who's watching the show, because the taxes you pay are the things that help me figure all this kind of thing out, and me and everybody else to figure these kinds of things out. And you have to sort of get people together to go on these cruises, because these oceanographic ships are real expensive to run. They cost like $40,000 a day for the cruise and the fuel and the so on and so forth. So you've got to make it worth it, so you load these things up with as many scientists as you possibly can, and then you don't waste a minute while you're out there. So it's sort of, you're working around the clock. As soon as you get to a station where you want to take samples, no matter what time of day it is, everybody runs out on deck and starts putting things overboard, collecting water, bringing water back on board, doing things in the lab, so on and so forth. So it's that's the general. I don't know if that's specific enough for the person's question. But.
Yeah, no, it sounds like a huge operation. So if you get to a spot in the ocean where you know that you need to take a sample at, and it's 3 a.m., you're out there no matter what time of day.
That's right.
That's incredible. Right. No sleep for scientists. That's
right. No, yeah. Sleep deprivation is sort of comes with the territory.
Yeah, definitely. Okay, great. So we also got a question about mercury as a marine contaminant. So this is kind of going back a little bit. So you explained to us how it moves through the ecosystem and how it can build in organisms, build in magnification or accumulation in organisms.
Right.
And so you also mentioned that mercury is a neurotoxin. So can you talk a little bit more about the harmful effects of mercury, and why do we want to know where it is in these huge concentrations?
Yeah, the health effects stuff is something that is not really in my field of work, so I can tell you what I've heard other people say. And if anybody wants to ask a more detailed question, I might have to just totally punt. But the most of the concern these days is that, so it can interfere with how neurons function. That's about the extent of how much I understand. And it most sort of insidiously is that it can affect how a person's central nervous system forms from the beginning if this person is growing. So you and I as adults are maybe less at risk for damage from mercury, but children and fetuses are most at risk because their central nervous systems are forming. So if we ate a lot of seafood and got some mercury poisoning, we might have some effects, but if we stop eating fish and let our mercury concentrations go down, which they will, if you stop eating, the amount of mercury in your body will go down over time. Has a half-life in your body of something like 6 months or something like that. So anyway, if you stop eating fish, then the amount of mercury in your body will go down. But if you're a child eating a lot of fish with mercury in it, then it might sort of permanently affect your central nervous system because the connections between neurons are being made and all that sort of stuff. You're building out your central nervous system still at that point. So that's all of the sort of public health rules about consumption and exposures and how you should try and limit the amount of fish that you eat and that sort of thing. Is really geared towards trying to protect children and women who are pregnant or women who are thinking about becoming pregnant, because it would affect their fetuses. Yeah, babies.
That's great. Well, I mean, not great, but thank you for answering that question.
Yeah.
So is mercury, I know it's a contaminant, but is it ever needed for something in the ecosystem that is technically a good thing? So is there a reason why mercury is needed in maybe lower concentrations?
Right. So it's a contaminant, but it's also naturally, of course, it's naturally occurring. It's an element. It's just out there. So it's we didn't make any of the mercuries, but what human beings are doing is that we're moving it around much more in the environment than it would be if we weren't doing the things that we're doing. So just to clarify the term of contaminant. So we do talk about there being anthropogenic mercury, but it's not. We didn't create it. We just threw it up in the air and got it moving. That's how we affect it. So it's sorry. And the other part of the question was, is it ever needed? I've seen one paper that maybe suggests that. So a lot of organisms, including us, need really small amounts of certain metals. Like if you look at your vitamin bottle, for example, you'll see zinc and selenium and nickel and so on and so forth. So there's some suggestion that maybe tiny amounts of mercury are useful for phytoplankton during photosynthesis, but it's the amount that they need is probably so tiny that the amount of mercury that's in the environment is way more than they would ever need. So you could, all the extra mercury that we're mobilizing in the environment is not helping in any way. It's at least that we know of so far. But
yeah, definitely. Okay, so Carl, we only have time for maybe about 2 or 3 more questions.
No worries. Wow.
And one of the questions that we received is when you are looking at mercury in the ocean or analyzing an organism, when is it a cause for concern? So is there a certain threshold that you say, oh, there's X amount of mercury in this ocean or in this lake or in this organism, we need to be concerned? So what is that threshold? And then how do you go about maybe fixing that?
Yeah. Fixing it is a big question, and I don't know actually really how to sort of fix it. So the cutoff, there's our public health sort of things, and if you're a person who fishes, you can go to your local fish and wildlife kind of thing, or your local public health or environmental protection agency or something like that to see these guidelines. There's a sort of typical number, which is if your fish concentrations of mercury are over about 0.5 part per million, so 1/2 of a microgram per gram of fish flesh, then maybe you shouldn't be eating that fish. That's not maybe the best way to talk about a person's exposure. It's really not what the concentration of the fish is, it's how much of that fish you eat. So this is the way that public health officials have sort of settled on a relatively easy way to try and communicate risks to people. And so, of course, you can't look at a fish and just tell how much mercury it is in it in general. So they often also just provide lists of fish that you probably should avoid eating because they tend to have more mercury. And not surprisingly, it's top predator fish, tuna, so on and so forth, sharks. And then also, just in general, older fish tend to have more mercury in them than younger fish. So when in doubt, the mantra is try and eat low on the food chain. So forage fish kind of things, anchovies, sardines, so on and so forth. And fish that feed and live on the bottom tend to be have lower mercury than big pelagic fish like tuna, etc.
Yeah, awesome. So throughout your research, have you seen mercury contamination or pollution to be much greater concern now than previously? Have you seen any type of, either is it increasing, decreasing, is it about the same? What have you observed?
Yeah, so there's been a dramatic increase in the amount of mercury that is out and about in the environment these days. That was part of what we showed with these sediment cores, and other people have shown with these sediment cores. So there's about we think somewhere between 3 and 4 times more mercury in the air and the water now than there was before sort of the Industrial Revolution. So that's right there. That, maybe that doesn't sound like a huge amount, but that, in order to get the whole globe to triple, that means we've put a lot out there. And it means now also that, presumably anyway, and we're trying to demonstrate this with real data now, is that fish probably have about 3 times as much mercury in them now than they did 200 years ago or something like that. The good news is that so a lot of the sources, a lot of the mercury entering the environment from human activity comes from burning coal. There's also a pretty big source of mercury from gold mining. And actually, the history of California is intimately connected with mining gold. And there was there was more mercury mined in California than there was gold mined in California. And as a result, San Francisco Bay, for example, has this legacy of very high mercury now stored away in its sediments as a result of mining in places like New Idria and New Almaden, just right nearby here, for example. So anyway, but the good news is that as a result of us human beings now burning a little bit less coal because other fossil fuels are cheaper, et cetera, and also emissions controls going on coal-fired power plants, the amount of mercury going into the air now, most people think, is starting to decrease. And it's maybe showing up in some places, the Atlantic Ocean, for example, which is much more surrounded by countries that are sort of decreasing the amount of coal they're burning. Maybe is actually its mercury concentration is actually starting to drop. Whereas the Pacific, we think, is still going up because places like China are still burning lots of coal and actually burning more now than ever.
Yeah, well, you know what? I am glad to hear that there are promising signs, especially over on the East Coast. So that's really, really great news to hear. So, and also I have one last question for you, hopefully more on the positive side of things. But Carl, what is your favorite part of this job of being a scientist or a professor? What do you absolutely love and is the motivating factor for you to do what you do?
Yeah, that's a great question. I think, I just like being outside. I think, and that's a fun part. It's nice to be able to have your work be involved in travel and being outside and that kind of thing. But it's really the part I think I've enjoyed the most, probably in the end, is just sort of working closely. You can't do this. This is not stuff that you do by yourself in the lab, just sort of with your head down. It takes a big army of people to do that, and it's always been really fun to be part of those teams and going out. It feels like you're conquering a mountain or something like that when you go to sea and you load all the equipment on there and everybody's sort of working together on one big common goal. That's a lot of fun.
That's awesome. Yeah, it sounds like there's a huge amount of teamwork, and it's always nice to go outside and learn something new about our environment. So that is so great. So Carl, I have I could talk to you for probably the next 6 hours about your research and mercury in the ocean, but unfortunately we are out of time. But do you have a place online, like a website, social media, or anything like that, where folks can go to learn more about you and your research?
There's a page. It's not very, this is something I've only been in Santa Cruz for a relatively short period of time, and putting up a website is just something I haven't really done yet. But there's some information about me and my lab at the UCSC Ocean Sciences Department. So that would be the best place to go. Yeah.
Awesome. Well, I will go ahead and put that link in our video description right after our conversation. And I'm sure it's just great. So it is, you're out in the field studying things that we definitely need to know about, and that's important. So, okay, well, that's great. And before we officially sign off, of course, I need to send another big thank you to everyone that tuned in for our conversation today. It's really supporters like you that keep these types of online programs running with the Seymour Center. So if you can, I ask that you please consider making a donation to the Seymour Center or becoming a member with us. You can always find more information on our website. That website is seymourcenter.ucsc.edu. You can also click on the link below in the video description. And while you're on our website, go ahead and sign up for our e-newsletter as well, so you'll never miss an upcoming program or event or a distance learning resource, and you can be notified of all of our upcoming activities. And so with that, I hope that you are able to join us for our next Labside Chat. For those of you that have been following along with this program, you know that Labside Chats are usually on the second and fourth Thursday of every month. However, the Seymour Center is going to be taking a break for the Thanksgiving holiday. So make sure to mark your calendars for the next Labside Chat on Thursday, December 10th. And more details will be announced on our website very, very soon. So thank you all so much again for tuning in. Carl, you are so fantastic. Thank you so much. It's been such a pleasure. And we will see you all next time. Bye, everyone.